Good morning, Gary Ritchie here with Pharmaceutical Manufacturing Magazine. I'm here at IFPAC 2009, and I have the opportunity this morning to talk with Dr. Brian Rohrback of Infometrix. Brian, for some time, has been the leader of an initiative called COPA, and I thought I'd ask you a few questions about the development and trends going on in COPA. COPA means...
COPA is Chemometrics for online process analytics, and the idea started back in the year 2000. Actually started in an Analect users meeting to deal with its main purpose was to deal with some of the discrepancies that existed among Chemometrics software and approaches to Chemometrics, and to try and give some sort of standardization to the world. In 2001, it was turned into a CPAC, Center for Process Analytical Chemistry initiative based out of the University of Washington.
That's a little known fact. Everybody talks about CPAC coming first, and now you're saying that COPA came before
Actually came before CPAC, but what CPAC did was they increased the purview of COPA from being strictly a MATLAB environment, which is what it started out for 1 year, into being a more general initiative to deal with improving chemometrics so that it could be used in process analytical environments. The difficulty was really that you would have all these different software packages, but then when you went to deploy that in an online process analytical mode, you wouldn't be able to use a model that you generated somewhere else. There'd be some difficulty. You couldn't read the file, or you had trouble processing a particular model, or different people would do things in different ways, and they couldn't make something that was proven in a laboratory work on a process. And so the issue behind COPA was to make that a uniform environment so that it was very easy to take something that was developed in a laboratory and move it online.
Sounds very much like what is being executed now in terms of quality by design and implementation. I think one of the things that came up a few years ago out of IFPAC was this concept of IFPAC MA, was to find a way to devise common platforms for all of these different types of analyzers and monitors. You're saying that the same concept could be applied to the chemometric software as well.
Exactly.
Wow.
So what was attempted at the beginning was to take people from chemometrics companies, so all the chemometrics companies were involved, and a lot of the analytical instrument companies as well, and the end user community, and put them into one room, lock the door, supply them with beer, and make it so that they could
the great
develop
lubricant of industry.
The great lubricant of industry. Well, they're just chemists, come on. But then to really sort through what the problems were and set up a dialogue. And probably for the first 3 years, the issue was mostly dialogue and very little action. But after that, we went through a process where the individual companies went away and started to develop products towards this standardization or in support of the standardization effort. In my recent recreation of myself, I recently was at United States Pharmacopoeia. Yes.
I had the opportunity to work on a project where I actually addressed this issue of different vendors' equipment, different software. I took a common data set. We investigated if you could actually answer the question using the same data set across different instruments and software, could I derive and arrive at an identical model?
Yes.
And I got to tell you, you can't.
Yeah.
And so you're illustrating exactly what I experimentally, and this will be interesting to see how in the future you're going to address some of these issues. What is going on with the developments in your company, for instance, in Infometrix? What do you see going on for you to help facilitate that?
Well, we've got a couple things that we would like to try and do. One of them is to take, as you mentioned, instruments from maybe different manufacturers or slightly different procedures for following those instruments, and make it so that the data that is generated by an instrument could be used by some sort of a universal model.
Yeah.
We've approached both spectroscopy and several types of spectroscopy, optical spectroscopy, NMR, and chromatography. I think of late, most of our activity has been in the chromatographic realm. And that turns out to be a much simpler task than spectroscopy in terms of standardization. So it looks like it's pretty clear that you can take chromatographic data that is coming from virtually any source, and you can align it to other chromatographs and use a single chemometric model or suite of models to be able to do predictions. Are you referring
to the what is this, ANML? That came out of ASTM. Some of the NIST people and some of the Europeans got together and created something along the lines of a common computer language, XML. This is ANML for analytical data. Is this the same thing?
Well, not really, because what it is, although everything works together, so that's nice. Anytime you have a file format standard that can be put in place, and Animal will provide you with that standard, the AIA, Net CDF style issue, those things are all file format standards. That makes interchange of data among instrument systems and for software suppliers much simpler. So that's one thing. The other thing is that there are some chemometric techniques that allow you to adjust chromatograms so that you can eliminate retention time variability, because every chromatograph has slight variations in retention time. That can be eliminated mathematically, and by doing that you have a way of having standards that you can make available for comparisons, for either classification or general pattern recognition of chromatographic traces. So that is a technology that's absolutely critical for this. So we have two things. One of them is the file format standards makes it easier. The other one is that if you can align chromatographic traces, then you can standardize them to what your expectation would be. You gold standard chromatography.
The actual signal and data itself.
Yep. So that's all signal processing, and that allows you to do automated interpretation of chromatographic traces. The same thread can happen in spectroscopy, optical spectroscopy, NMR, any analytical technique, X-ray fluorescence if you want. So any of those techniques, you have the same steps: a signal processing step, being able to read it, process the signal, and then do some form of pattern recognition.
Well, Brian, this is certainly going to be important in advancing this idea. Where we are this year with IFPAC 2009 is the idea of implementation of QBD. This discussion is an illustration of the actual nuts and bolts and details of part of that implementation. I want to thank you for discussing this with me today, and it was a pleasure to see it.
It's always a pleasure to see you, Gary.
Great.